Hi, this is Dr. Ben Morrill. Welcome to episode 76 of Reptile Genetics Weekly. Glad to have you here. It's been busy, and you may catch it a little bit. Both Kayla and I are getting beat down a little bit by the bugs going around, but we're here. We made it. For an update for samples coming in, we will have fast results going out pretty much every day this week. We do have all of them going from the new 10 that we have. Those are all going as fast as well. And so if you send in any for those, you should be getting those as well last weekend, this week. Sex determination's going out a couple times a week. And then the panels, we had results go out last week. And so if you didn't get results on that, the next ones, my guess will be coming in about a week and a half, something like that. We'll be doing the next panel run. So it's coming. It might be 2 weeks, a week and a half to 2 weeks, something like that. We'll have the next panels coming. So, but yeah, I think that's all I have for sample updates. We have several other things we'll talk about, but we'll bring Kayla on. How are you doing?
Hey, Ben, I'm doing well. I got the sniffles, but I'm okay. Like I said, I'm going to hit some Zicam, and I'm getting my flu shot and everything. So if I have to mainline Dayquil when I get to Collaborator, I'll mask up if I'm still funky, but so y'all stay hydrated out there. If you got symptoms of some sort of funk, you might want to mask up when going outside, but outside in like public mingling with people, but 'tis the season. It's great. But thankfully I think I'm over the worst as of a couple weeks ago. So,
I felt good while I was in Charlotte at the reptile show, but yesterday when I woke up and then today again.
Oh no.
Best, but I'm hoping like you, I'm very excited for the Collaborators Fest this weekend. So I hope I can get feeling better by then.
Yeah, of course. Despite feeling the funk, I heard you were on a podcast recently, right?
Yes, thankfully he recorded it before I was feeling sick. So
good.
Good on
that. Yeah. So tell us about it. It was that Wolfie Royals. And is he in New Zealand or Australia?
I believe he's in the UK.
Oh, in the UK. Oh, okay.
Yeah.
Fair enough. Very cool.
Go on and talk a little bit. It's fun to kind of get a little bit different questions. They're in a different market and thinking about ball python morphs and genetic testing from a little bit different point of view. So it was fun being on there. We also talk specifically about like some of the history about desert ghost, and so we kind of do an overall overview of desert ghost and how we got to the point now where we only test for desert ghost C. So yeah, it's a good one. Tom's great and a good person to support. So I think he's on Instagram and YouTube, and he may be other places as well. So
yeah, definitely
good person to support.
Yeah, and I'll be sure to have a link in the description so you can go and listen to the podcast episode. And if you are one of the people who've been wondering about, you followed the DGA and B, like the DGA and B thing when we were first figuring that out, and then later on you saw how it turned into just testing for DGC. So if you're curious about how that ended up happening, that's a great overview. So well worth a listen.
And let us know in the comments if that's something you want us to talk about on this show. We did kind of go through all the details from the very beginning when we knew that we didn't know everything. We let you know we were going to look for DGC, and then we did find it and kept you updated. But I know those are now, like a year old. So
yeah, exactly.
If you want an overview on this show, let us know in the comments, and we'll be happy to do that.
Yeah, definitely. But just to knock out a few business things before we get into the meat of the episode. Hey, if you want lower shed testing, be sure to listen into this. Lower price shed testing, I should say. We'll just skim over this real quick just because, we hit it every week, but it's really, really helpful for you guys. We do this for y'all. If you have a lot of sheds that you want to test, but, the price is really high, the more snakes you test, shoot us an email. Shoot Sean an email at raregeninc@gmail.com. For morph testing, bulk testing can get as low as $23 per mutation test, or even as low as $15 per test if you're doing like bulk colubrid sex determination. So those,
and even then I'll throw in if you have way more, sheds, you got a ton, then it could even be lower than that.
Oh yeah. I mean, we got people who do exactly that. I mean, there are some folks who Sean gets a box from pretty much every week at this point, right? I think.
Yeah.
So it's definitely worth it. We want to give you guys the most bang for your buck. So yeah. And likewise, it doesn't have to be a ton of snakes, especially if you're doing those morph tests. If you're testing 4 snakes for 5 mutations each, that's still 20 tests. That counts as bulk. Shoot us an email, see what we can get you. And send a list of what you want or have a spreadsheet. Sean will give you a quote. So.
Yep. And we also talk about this most of the time, and I'll just throw in there really briefly. If you have one animal you want to do multiple tests on, you can also get a discount on that, either through Morph Market or Clutch. They have ways to bundle or group those. So you're not paying. So like if it's, 3 or 4 different $50 tests, you're not going to be paying $50 for each one. So those of you that only have a few animals and you want to test for multiple things, then we can get you a better deal on that too.
Yeah, definitely. And so tell us about Charlotte. How was that this weekend?
It was good. It was a lot of fun. Thank you to all of you that were there. And I really enjoyed walking around, saying hi to everybody and talking and enjoying the show. I realized on the way back, and then my wife always gives me a hard time. I'm not good at remembering to take pictures. And Kayla, one of the first questions she asks is, "What pictures did you take?" And yeah, I forgot. Pictures. I got to get better about that.
That's okay.
But it was a lot of fun. I did want to show some of the stuff. So from Balls and Strikes, Ball Pythons, Margaret Woodruff. So this is a painting I got from her.
Oh, that's so nice.
Yeah. Oh, so it's a banana combo. I believe it was from Leviathan snakes that originally hatched it out. So pretty cool. And then this is the one that she...
Oh my goodness. I love the reds in that.
Oh yeah. For that one, she got the pattern so well that when Justin Kabelka, this is a Canova animal, and when Justin Kabelka was walking by their booth, he stopped. He's like, "I know exactly which animal that is."
Oh, that's awesome.
Yeah, pretty cool.
Beautiful work.
Yes, and I don't have to feed and clean up after those, and they still make me happy. So thank you, Margaret.
Oh, great job, Margaret. Those are fantastic.
But we have my kids actually have been working with crested geckos.
Oh, we got a new buddy.
Yeah, so from JLP Morphs out of South Carolina, that they've been sending us lots of pictures when we do shows. So we did get another crested gecko.
Look at him. Oh, he is precious. What morph is that?
I know he's Harlequin. I don't know what else. I don't know the crested geckos very well.
Yeah. Oh my gosh, he is adorable.
He's handsome.
Yes, he is. Does he still have his tail, or is he a frog butt?
He does.
He still has it. Awesome. Yep. I mean, granted, I love frog butt cresties too, but it is kind of a I know that's always a concern with some species that drop their tails. It's like if you ever have a legless lizard or something, like that's a lot of animal with a tail you can lose.
Oh yeah.
Makes me nervous. However, love them either way. And I know they just decide to do it sometimes. It doesn't even have to really necessarily be a stressor caused by you. But that's awesome. And you said you guys hatched out your first ones, or your kids hatched out their first cresties recently, right?
So this is the one from the first clutch that we hatched.
Oh, look at!
So
oh my goodness!
He's thinking about. Oh, there he goes.
What a cutie! They've already gotten. Good jump.
Yeah.
Go, little man.
Things.
Look at him. That's a good baby.
So yeah, they're fun little things.
Oh my gosh, that's awesome. I love that your kids are getting into reptiles and breeding their own critters too.
Yeah. It's fun stuff. But yeah, I really enjoyed it.
Good.
Both Nick and Lauren and Troy and Lisa, I mean, they were working hard. They had some some various things to overcome. I think the show turned out really beautiful. There's lots of people from lots of places. I've gone to lots of Charlotte Repticons, and this felt really nice. My wife and kids came, and they've gone to Charlotte Repticons too. And as you would imagine, a show like this is much nicer. They really enjoyed it. They got some reptile related jewelry and
Ooh, fun.
things. So
Very
yeah,
cool.
it was a really nice show, and definitely some very cool animals there too. It was awesome.
Yeah. Well, awesome. Sounds good. Glad you guys had a good time. Glad your family got to tag along and get some cool knickknacks. So that's great.
Oh, yeah.
And then this weekend, we're going to Collaborators Fest up in Indianapolis. And we are sponsoring Collaborators Fest. So me as the resident Collaborators nerd, I am so hyped for this. Flight's ready. We're going to be up there, and you'll find our booth. We will have a shed drop if you want to bring us some sheds, not just for colubrids, but also whatever you happen to be breeding. Or if you have ordered a test and you have it labeled with your order number, you can drop that in the shed drop box as well, or hand it to one of us, because Ben and I will both be there.
Yeah.
So really excited. Yeah. Anything else about Collaborators Fest or show stuff?
Nope,
That we
I'm
can think
looking
of?
forward to it. Yeah, that's going to be cool. It'll be the first time that I know of there's a show where everything that will be out there will be colubrids. It's all colubrids.
No, it's going to be so cool.
Yeah.
A huge variety. So I'm excited. Under or over, you think I'm going to walk out with an animal, because it might happen. I'm determined not to, but
you have a tough time not getting something.
Oh yeah. Well, anyway, let's ooh and ah at some cool looking ball pythons, because we're talking about the Hypo gene today. And it's time for our monthly-ish Morph Spotlight. So let's talk about some hypos. So this animal actually we featured on last week's show, but it's a hypo in addition to a Fire YB GHI Powerball hypo clown. It's just so cool looking. You can't not look at it. It's awesome.
Very cool.
Of course, that's one of Kenova's creations. Let's see. We've got Balls of Fury, a GHI YB hypo, which I love. I just can't. I love hypo with dark genes. It looks so good.
Me too.
And we've got from JLP Morphs, a chocolate butter hypo who's feeling very shy. But I love the little, like, not quite highlighter yellows, but like those really high saturation yellows that are like peeking through around the dark outlines of the markings. Really nice. Let's see. And we've got this hypo clown, which it's funny. I sometimes forget what just a plain clown looks like sometimes because I see so many combos with it. Not that that's a bad thing, but that's just like a really, really nice pattern. I can see why people love it.
It's pretty.
Odie Enchi hypo, also from Balls of Fury. A whole lot of folks sent us in some pictures of hypos. So really nice looking. Another JLP, a pastel butter hypo. Let's see. On the left, this is from Inky Clouds, a mahogany hypo, and the one on the right looks very familiar because that's one of an animal you produced, isn't it, Ben?
Yeah, that's one of Cisco's babies.
We love Cisco.
I never would have guessed that much orange would come through. That they look like just like orange cream ball pythons. They're crazy.
Yeah, and his dad, like if you look on his head, there's kind of like a grayish or bluish look there. His dad, Cisco, like he almost looks in a good way like moldy cheese. And so as soon as I saw this snake, I was like, oh, that's a Cisco baby. It's gotta be. And sure enough. So it's always nice to see little updated pictures of animals that you produce and also that I've met by being in your snake room.
Yeah, you took the picture that was in the ad that Tommy saw when she got that.
That's right. Yeah. Let's see. Black-Hood Mojave. I really like the contrast. I think I like yellow and neutral tones together because whenever I see that in a ball python, I'm like, oh, that's pretty. Black pastel hypo from Balls and Strikes, an OD hypo pied next to a redhead hypo clown. Does redhead actually produce like a reddish head or like affect the head pattern? Because that one actually does look like it has like a little, it does look like it has a little cap on it, on the redhead animal.
Yeah, I honestly don't know a lot about redhead. I know it's a very popular mutation right now, and one that we're hoping to get more sheds from. So any of you that have redhead or redhead combos, send those sheds in, and we'll make a test. But
yeah,
I know they make some really cool combos.
Yeah. If someone wants to explain in the comments to the colubrid person what redhead does in ball pythons, I would be very cool with that. And yes, send us sheds.
I want to say that it's also the name of the people. I think it's redhead reptiles.
Oh, okay.
For sure. Someone can tell us in the comments which part of what we said is true.
Yeah. Sounds right. Spot Nose Fire YB Hypo from Armless Angels ball pythons.
That's awesome, Justin.
Great. Armless Angels is that's a really fun name. It's kind of like I forget whose tagline it is, but there's a breeder out there whose tagline is legless excellence. And I've always like loved that. So that reminds me of that. And Hypo DG.
So nice.
Great picture.
Yeah.
Nice and blue, and also just great pose. Like that is a sweet little loaf of a snake. And I love this animal.
Yeah.
Super OD. Enchi YB banana hypo pied. That's man.
Yeah, that's right up my alley. I love orange in pieds, and that one, the hypo on top and banana in there. That's really cool.
Oh yeah, like that's like an orange sherbet swirl. Is it sherbet or sherbet? I don't know. I can't read. It's fine. Either way, that snake looks positively edible. And we got a nice hypo chocolate. I think chocolate and hypo are both really nice together. That's a good gene combo. From Wetter Morph Constrictors.
Beautiful.
All right, so let's talk about the mutation itself. Mr. Geneticist, take it away.
Yeah, so this is one. There's only one mutation that we test for, and that the other companies that do testing, as far as I know, there's just one that we found. It does seem like there are at least one or two other lines that are not very common. So really, the vast majority of hypo ball pythons, whether they're called orange hypo, orange ghost, or hypo or ghost or whatever, even the yellow ghosts from back in the day, I remember some people had yellow ghosts. They seem to all be this same -5 mutation. So there's 5 A's, T's, C's, and G's that have been deleted from the 8th exon in the MLPH gene. So it's a -5 deletion. It gives us that cool look that we see in all these hypos ghosts.
Very cool. And so the paper that it came from was this one, which I will also link in the description if you want to read it. What year did it come out? Because it's been a couple of years now, I think, right?
I think so. I think within the last year or two, something like that.
Yeah. So in each of these figures, like what are we looking at?
Yeah, so this is work done by Dr. Hannah Siddell and her students, again, Eastern Michigan University. In A there, you can see a normal ball python and then what its shed looks like. And then in B, you can see a hypomelanistic or ghost ball python. You can see the reduction in the darker pigments. The hypo means reduction, and melanin is the dark pigment. So it's a reduction in dark pigment is hypomelanistic. And you can see that shed. There's very little pigment there. That's another way you can tell if an animal, if you're not totally sure, you can wait till they shed. And usually the hypo ball pythons, their sheds will be clear or almost completely clear. So
very cool.
Yeah, very interesting. And then in part C there, what the melanophilin gene does, MLPH, what that gene does is it helps be part of this apparatus that moves melanosomes, or you can think of it as the dark pigment, the melanin itself. It helps to be able to move that dark pigment around in cells. And so if there's a problem with that green part there, that's the melanophilin, then that way to move the dark pigment around gets messed up. So that's like, needing to have a car to transport you around, and then all of a sudden there's no car. You're not going to get transported. So that means that there's no dark pigment that's moved out into the tissues. And so we have that reduction in melanin just because it can't get transported out. That's the part of the process
Oh,
that's getting messed.
interesting.
Yeah. And then in D there, that's showing that eighth exon. So if you count out there from the left, that's the eighth exon. That's the part of the DNA. An exon is the part of the DNA that makes something that actually becomes part of the protein. So it codes for amino acids that become part of the protein, that becomes part of the melanophilin protein itself. And that -5 in that eighth exon causes a frame shift. So that makes it so all of the amino acids after that are messed up, and a lot of the time will cause an early stop codon or cause that a truncation. And so you end up with a protein that is not the same as normal, and in this case it makes it so it can't transport that melanin anymore, and therefore we don't have as much dark pigment.
Okay. So with when those five letters or proteins are deleted, is it where a gap is left in there, or does it move everything around it up because it's been deleted?
The second thing you said, that's why it's called a frame shift.
Okay.
So there's two different sequences that are important. There's the
Yeah.
DNA sequence, and that's the nucleotides. That's the A's, T's, C's, and G's. So there's 5 A's, T's, C's, and G's that get deleted.
Yeah.
Then the second sequence that's important is the amino acid sequence, and that's what actually makes the protein. And so every 3 letters of DNA, so like an ATG, that's called a codon, and that makes a single amino acid sequence. So ATG is a methionine start. So ATG in the DNA equals just an M or a methionine in the amino acid. So really, we're cutting out one full amino acid and then 2/3 of what would code for another. And that's why it messes up the frame, because there's 5 that are taken out and every amino acid is coded by 3. And so it messes up the frame for everything else after that.
Oh, okay. Cool. Thank you.
I don't know that was actually helpful, but
actually, no, it did actually. That
good.
cleared up for me more. So thank you. And that also helps explain. Oh wait, no, we didn't get to every one of these actually. So please continue.
Yeah, so E, you're seeing the sequence of the DNA is truncated, or actually that's looking at the amino acid sequence. So the melanophilin is 598 amino acids normally. With this -5, it's going to truncate the protein. You only have 363 amino acids, so it makes the protein much smaller and non-functional. So, and then the F down there, that's looking at all of the samples that were run in this study. So, you're seeing 5 ghosts, and you're seeing, I guess that's what, 130 or 150 non-ghosts. So they're either hets or they're not ghost at all. So
okay. For this next one, that does kind of explain what you were saying about, it's not able to transport the melanosomes.
yes,
because when I was looking at this earlier, you see under the wild type, here I can pop our little faces up there. Under the wild type, you see the little dots that are the melanosomes, or the actual pigment that we'll see are spread out throughout the skin around the melanocyte, which produces the melanosomes. And then in the ghost, and what they believe is happening there, or hypothesized to be happening, is the melanosomes are all produced there, but they're just mashed together in one spot.
Yeah. They never get transported out. There's no car to take them out to all the cells like in the wild type. You see those little dots that gives it the darker color. But in the ghost or the hypo, there's no transportation going on, and so that's why it's a much lighter color.
Interesting, because that reminds me of how like there are a bunch of different genes that in the reptile world we refer to as hypo or hypo-like. For example, like here I go back to my rat snakes. We have a hypo-like gene in rat snakes that is reduced, like severely reduced melanin, but it's not a fully amelanistic, but it also has no black on it. Like whereas opposed to the hypo gene in ball pythons, you actually see some black on them, meaning that it's just an entirely different function, probably, that reduces melanin. In this case, the melanin kind of gets stuck somewhere rather than being produced less, if I'm explaining that right.
Yeah, that's a very, very good point. So the melanophilin gene and the melanophilin protein, when you mess that up, you mess up transport, but the melanin's still getting made. But yeah, another potential pathway to get a hypo looking animal would be to decrease the production, and those would be totally different genes. So
yeah.
Yep.
Okay.
Definitely perfectly right.
That's really cool. Okay. So here's an example in what are they called? Modi dogs? The Modi dog breed. Which I believe is a Belgian herding dog. And this mutation, well, here, do you want to explain it, Ben?
Yeah, so the dark black one in A, that's kind of the normal coloration. And then there's at least 3 different mutations in the melanophilin gene that they've found in dogs.
Yeah.
And see that you kind of get very different things. You might end up with like D, which is kind of like our hypomelanistic ball python. That would be kind of like the hypomelanistic Modi.
Yeah.
Even other mutations in that same gene might lead to like some modeling or even like in B there you get kind of like a light brown and white, and so you get some different looks from these different mutations.
Yes, it is. I think F is the most, well, E and F are the most interesting because you have a very light colored animal, but with very dark skin, like lips around the eyes. The eyes themselves are dark, the dark nose. And so, usually when I see a light colored animal, I think, oh, the melanin production was either halted or severely reduced. But in this case, if it is the same mutation, it's actually that the melanin is there. You can see it right in that, like on F, its nose, its eyes, the melanin's there. It's just not being transported out to all the other places where the melanin would be spread out to.
Yeah.
Interesting.
I think in F, E, and F, there's another gene at play also where it has the EE.
Yeah.
So that's not only a melanophilin mutation, but something in another gene as well. So
okay.
So yeah, pretty cool.
Yeah. And then same thing, I guess, in these 3 puppies.
Yeah, these are Modi puppies. It's just showing them side by side. The 2 on the outside are normal coloration. The one in the middle is the hypomelanistic one. So pretty cool.
Very cool. All right. And then again in Mink, where we've got the standard dark brown, silver blue, and headland white.
Yeah, so in this case, you can see normal brown, you can see a hypo, and then you can see a leucistic. And so in this case, we're looking at the silver blue. That's a melanophilin mutation also that causes that look that they call their silver blue. And ball pythons, we call our hypo. So
interesting.
Yeah, in that case, they have a whole exon that's missing. So the whole exon got deleted. Instead of, we got just 5 base pairs, but in this case, they got a whole exon that got cut out, which is usually hundreds of base pairs.
Wow. And that's so interesting that it caused the same let me make sure I have my vocabulary right. Go back one. The same melanophilin protein, right?
Yep. Yeah.
Melanophilin protein that has stopped working.
Yep. Yeah, it broke it even worse with
Yeah.
that. But broken either way, it leads to a similar look. So
Oh yeah.
pretty interesting.
Cool.
So if you want to know what hypo in a ball python looks like in a mink, there it is.
Nice. Yeah, and these are all of the contributors to this study, which is a text wall, but it's also, a whole bunch of familiar faces.
Yes.
Like there's Dave Dunn of Ball and Hand Pythons. He's like our neighbor, pretty much.
Yeah, he's right here. Tammy Hutchinson, she's been on most, if not all of these. Michael Cole.
Yeah.
There's lots of Kyron Graves that works with Dr. Siddell.
Yeah. Ozzy Maldonado. Is that Ozzy Boyd's Ozzy or a different Ozzy?
So. I'm not sure.
Okay.
I don't think that's his last name, but
okay.
But yeah, this is a good time to say a shed donor shout out. In this case, it's the donors for this research. Thanks once again, like we say, and we don't still, even though we say it all the time, we don't say it enough. Thank you all so much for being willing to take the time to collect sheds and send them in. And, whether it's to Dr. Siddell or us or, Hannah or Heather up in Canada, it's so awesome to be able to get sheds sent in and get these things figured out, because just like we can't do it without you all, Dr. Siddell and any of us, we can't do it without you guys sending sheds in. So I know it's like Kayla said, a wall of names, but once again, thank you so much. It's awesome to be able to make this progress.
Definitely. Let's see. So because this, how many hypos have we tested at this point?
Yeah, definitely thousands.
Thousands. Yeah, we've tested thousands of hypos, and there's been a paper published about it. So we grade this an A+.
Yes.
And what does it mean what does that mean for people who haven't seen our grading system before, Ben?
So it's just a very, very well-established test, and it's not just us running it. There are other testing companies that run it, and as far as I've heard from everyone, this does seem to be the best test, and the one that does give you the correct results for the vast majority of hypomelanistic ball pythons. Like we said, there is at least one or two other lines that are not compatible, but they're a very small portion of hypomelanistic ball pythons. And any of you that do have what looks like very obviously a hypo ball python, and you run that with us and get a negative, we would be very interested in getting those sheds, because we can sequence and find out. It might be something similar to what Kayla was talking about. Maybe instead of messing up the transportation of the melanocytes or the melanin, maybe there's another gene that's been affected that makes it so not as much melanin gets produced. But anyway, we would love to be able to sequence those, even if it's a smaller percent of the hypomelanistic ball pythons. That's still something we're interested in. And I'm sure Dr. Siddell would also be interested, since she's done that paper. If there's any that don't have that mutation, send us sheds. But like I said, the vast majority of you that have hypo or are interested in getting hypo ball pythons, this -5 and this test is very good.
So now let's move on to a little sponsor love with one of our newest sponsees, Green Room Pythons.
Yeah, so Bob Bledsoe, he's a it's very lucky for us in the reptile community that has an interest, and he's a very awesome person to have be a part of our community. He has a background in acting and doing comedy. I think I remember him saying, I think it was actually one of the people watching one of his lives asked him if he was really in a episode of Good Luck Charlie, and my kids used to watch that. They were pretty excited to hear that Bob Bledsoe had been in Good Luck Charlie. So yeah, he's done some acting. He's a comedian. If you watch some of his videos, it's really nice. He has an entertaining way to talk about them, and he does a very good job of focusing on helping people learn, especially if they're new to keeping pythons. And it's not just ball pythons. He's got retics and black-headed pythons and, a few other species. And so it's really beneficial. It's a very positive, uplifting, and educational. But then also there's the entertainment side. He's got a twin brother that looks a lot like him that helps
Does
him
he really?
with some of his helps him with some of his shows, and that's pretty funny too, to watch him interact with his twin brother.
Oh, nice.
Yes, yeah, he does a great job. It's really good, really beneficial. And then especially any of you that have friends that are interested in learning about keeping and breeding and what it's like just recently Bob was moving, changing where he was living. I don't think he was moving very far, but he did some episodes and talked to some people in the industry that have had to move collections, and just kind of talked about some of the things to think about when you're moving and have live animals, specifically pythons. Think about and make sure you do. And I think an episode yesterday was talking about one of the snakes getting loose in his new place. So
Oh no.
I didn't watch it yet, but I'm sure he's talking about how to. I would imagine anyone that was new to snakes at least once or twice had something get loose. I certainly did early on. Thankfully, it hasn't happened in a while, but
I've had it happen twice. Okay, 2.5. Two and a half. The
half.
half. Okay, to explain the half. The half. I don't know how this happened, but I think I left like the sliding door to my boys club, as I call it, for my male rat snakes, open like this much, and somehow Mr. Lemon Bars of the fingers following fame, this goober, he's like 5 foot long now. He's like this thick around. I don't know how, but he managed to squeeze out of there. And I didn't realize until I was sitting right here in this chair and I heard tail buzzing. And normally that's really unusual. My snakes are super chill, and I was like, something's wrong. I walk over there, and Lemons is furiously trying to get into his tank on the outside of it. He is like nose rubbing against it, like, oh God, I have to get in. And the cat's just looking at him. And I'm like, my son. So I pick him up. He's fine. The cat hasn't messed with him. Honestly, he could hold his own against the cat at this point. But even still, I do not want to risk that. He was fine. Just very upset and smelled like he had musked a little bit, but
Yeah.
I don't know how. It was so stressful. But I think he managed to squeeze his way through there. And like the tank, the sliding glass door was not open anymore. I don't know how, but scary. So back to green room pythons.
Yeah, so very valuable resource. He's doing a great job, and he does lots of lives where people are able to ask questions, during the live and get a live answer. So it's a really, really nice, and he's certainly earned a very healthy following. I think he's got, tens of thousands of subscribers. That's awesome.
Yeah. 67K subscribers. He's made over 321 videos. So
very cool.
Yeah. Also, I love his logo. We were talking about that before we filmed, but it's got like the snake head with the scales that make a G. And also it looks like a guitar because he's also a musician. It's great stuff. And he is on Facebook and Instagram as well, at Green Room Pythons. And he posts pictures, posts snake stuff. Snakes and Scotch, I think, is his live stream name, which sounds like a great time. If you partake, and even if not. So he's good people. Definitely recommend checking him out. And we're very proud to sponsor him. So yeah, I think that's all we got today. Got anything else before we call it a day?
Nope. Just thanks again. Make sure you do all the things to help the algorithm get this video out to more people. Like and subscribe. Give us comments. Let us know what you're interested in seeing, and if you want to hear more about the DG saga and anything else, we do read the comments and appreciate them, and it does help get our information, our videos in front of more people. So that's a way you can directly help us grow the show.
Yes, please. And grow the show helps us pay the bills and also goes toward making new tests. So Thanks, guys. Hopefully see you at Collaborators Fest this weekend, and we'll see you next time. Outro in 3, 2, 1.